I would like to about you, because indeed, from what I understood this morning, you are quite a diverse audience. So I thought maybe I can ask how many of you are veterinarians? Okay. How many are studying to become veterinarians? Okay. How many maybe have koi as a hobby, or you are culturing koi? So the ones that are not veterinarians, which were, let's say, 60%, what are they doing?
Okay. And
Okay.
then lecture to mow and...
Fish, when the virus will pop up out of a host cell, will look, of course, like a virus, but it will also look a little bit like a host cell, cells of the host. In particular with Koi herpes virus, it is the largest herpes virus genome which has been sequenced so far. Colleagues at the Veterinary Institute in Lelystad. I've heard that we are still lagging a little bit behind with the health certificate for this fish. From what I understood, and that's actually when I'm looking at you, some proteins and put them on its coat, but it has also stolen some genes from the host and also incorporated them in its own genome, and more as they should be. Therefore, they will not end up killing these cells anymore. At the same time, so not later, not after the infection has occurred, but at the same time that the virus is interfering with the activity of this TNF, it's also inhibiting the cells because it has its own IL-10. So this we came to know because in 2005, found out what's the infection route of a well plate. And the virus now, we have identified the virus, we know all the symptoms. Some people, of course, manage to come up with some solutions, but we do not actually know what's going on in the fish. We have a machine to record the fluorescence on the body of the fish or eventually on the organ of the fish. And as you can see here, it's basically spread all over. And you flip it over, it's here, you take this part, it's here, you take this part, it's here, and then you take the fin. You can see that basically in the organs, there's absolutely nothing. And in the gills, there's absolutely nothing. If I zoom even further, you can see there's some virus here on the skin of the mouth, but there's absolutely nothing in the gills. They sent first the paper to the reviewers, just like this. These were the tube, and the virus was just provided to the water. So here the virus could go through the gills or through the skin, could go anywhere. Well, in the lower part, what they did, I'm not going to say exactly which rubber tube they used, but they used a rubber tube to isolate the fish, or let's say the back part of the fish from the front part of the fish. Here, there is an O-ring, so it's tight. And you can see that there is no leakage from this side to this side because they kept the height of the water on one side of the tube higher than the other side of the tube. If there would be a leakage, or if there would be communication between the two tubes, then the levels would be equal again as the virus. Conventional vaccines, for example, inactivated vaccine or T cells, so the ones that will remember that they have seen a pathogen already and they will respond faster the second time when we see it, just freely swimming around, and therefore they will produce antibodies and they will neutralize the virus, so the virus cannot attach to a cell, enter to a cell anymore. But most importantly, we have these macrophages. These macrophages are the cells that you basically hear all the time, and they are a little bit everywhere, but they are really, really important because they produce these so-called cytokines, these messenger molecules, that will tell to the B cell, please proliferate, will tell to the T cell, please, now kill. It's just in the presence of these cytokines that all this occurs. So I would like you to remember this part because I will come back to that later. And I will remind you again to read it again. So in lab scale, it protects 100% of the fish. Now, I already started mentioning, it has only 2 proteins on its surface, and the protein G is the one against which the antibodies are made, neutralizing antibodies are made, and they have been starting this work already like 10 years ago. What they found out is that DNA proteins of the virus. So we have mimicked the natural infection route of the virus without actually having the presence of the virus. So injecting this DNA into the muscle causes the plasmid DNA to go into the nucleus of the cells. Then we then use the machinery of the cell to produce the protein. The protein is expressed on the surface of the cell. Then there is a cytotoxic T cells passing by, there is a macrophage passing by and says, hey, this protein doesn't belong to you, I'm going to kill you. Of antibodies, memory T cells, and memory B cells, which is what we said in the beginning, all that is at the basic of vaccination. We performed experiments, and these have been done a little bit to somewhat larger scale, so here we are talking about 300 to 400 fish being injected all the time. It is infected with spring Barrichello virus, and without going too much into details of the immunological part, this was again the last question.